what you need is to get C15 integrated into the cell membranes for these long-term benefits and into our cells and into our tissues. So that can take about 3 months.
Welcome to Peak Human Labs, where I share amazing interviews with incredible human beings who are leaders in how to optimize your health and get you to peak mental, physical, and mental state. My name is Dr. Sanjeev Goel. I'm a physician for the past 20 years, specializing in longevity, precision medicine, and biohacking. And my mission is to alleviate the suffering we all face and will encounter when dealing with illness, sadness, and loss. And to this end, I'm sharing these conversations with people who can shed light on how to live more purposefully and intentionally so that you can be inspired and create your own path to wellness and fulfillment. Hi, everyone. Today we have Dr. Steph from Fatty15 on the podcast, and we did talk with her last year. It was a huge success. People loved knowing more about this incredible nutrient called C15, and she's got a lot more updates, a whole new book out called The Longevity Nutrient. You can purchase the supplement at fatty15.com/Dr. Sanjeev Goel. We will talk to you on the other side. Okay, take care. Hi, Dr. Steph. Great to see you.
Hi, Dr. Sanjeev. Wonderful to see you again.
Yeah. So I think we chatted about a year ago. We did a podcast episode and we talked all about Fatty15. We've had great success from our patients who are using it now. And I think a lot of people online as well got more familiar with it. So how are things going with C15 since then?
Great. Keeping busy. Awareness has increased greatly, in part thanks to you, Dr. Sanjeev. So a lot of conversations, a lot of people learning and wanting to learn more about C15 wonderfully since we last talked. Just the research around C15, over 100 peer-reviewed studies, to date and growing, and then which then resulted in Simon & Schuster reaching out and saying, "This seems like there's enough for a book." So that's hence the coming out with the longevity nutrient, the book that came out earlier.
Yeah, so let's talk about the longevity nutrient and tell us about the book. What really were the key messages of the book, and how did obviously the longevity nutrient, I believe, is C15. So tell us about that.
That's right. I know that would be a surprise, right? It's something else. People who know me now, it's like, that's what she talks about. So with regard to, we basically wrote the whole story. So the book covers, this amazing, surprising and accidental discovery of C15, which is also called pentadecanoic acid, an odd chain saturated fat, right? And so the discovery that we made accidentally in dolphins, right, while taking care of older dolphins, we go do a deep dive, forgive the pun, into that story, but then also really just show the breadth of studies and science and robust support of the relevance of this nutrient, right? Emerging as the first essential fatty acid to be found in over 90 years since omega-3, and really covering the science, right? From the oocyte, right? From fertility all the way through our latest years. So was really excited. We talked about, nutritional deficiency syndrome, the first to be discovered in 75 years, and all of it, just tons of science, hopefully told in an engaging way to not put people to sleep, but instead to inspire a movement to help get C15 back into our lives through agricultural practices, food changes, all the things.
Okay, so let's now do a little recap for the new listeners who haven't heard the previous podcast, but maybe your background and how you were involved in dolphin research at the very beginning, and how you got acquainted with C15, because you're right, before you, I had never heard even of this fatty acid, let alone understood that had any health implications. I mean, I understand there's different fatty acids for each of different lengths of carbon lengths, but I didn't think anything special about this one. We all hear about omega-3. Yeah, so maybe just take us through that.
Absolutely. And that was the whole world. I mean, really kind of the same with even experts in fatty acids, the same space. So, so hi, everyone. I'm a veterinary epidemiologist and was recruited by the Navy 24 years ago to help continually improve the health and welfare of aging Navy dolphins, which was wonderful. I thought I would do that for a couple of years, and it ended up being a couple dozen years. And it's really an incredible population. The Navy has cared for this sustained population of about 100 bottlenose dolphins for over 60 years. And because of their great care, Navy dolphins live a lot longer than dolphins in the wild. So dolphins live to about 20. At the Navy, they're living into their 40s, 50s. And I just learned, I went to go visit the crew just last week, and I learned that 2 dolphins this year are going to turn 60 years old.
Oh my God. So it's like that in human years would be like a 150-year-old dolphin.
Yeah. So amazing. And so
what's the limit? What do we think?
Boy, they're pushing it. We're gonna find out. So
do these 60-year-old dolphins are metabolically very old?
So it depends.
They're looking really young here.
Yeah, well, and that was the whole interesting finding. So I was brought in to understand aging dolphins because there's no knowledge of it because they don't typically live that long in the wild. So what we found over a 10-year period, over a decade of studies, is that about 1 in 3 older dolphins develop insulin resistance, high cholesterol, chronic inflammation, fatty liver disease, right? This is all sounding very familiar territory. And as we just published a couple of weeks ago, even the full suite of changes consistent with Alzheimer's disease. So we were able to leverage this advanced technology called metabolomics to look at thousands of small molecules present in their archived serum, as well as their all fish diets, to see which small molecules predicted the healthiest aging dolphins. And that's where we thought it would be omega-3s because all they eat are fish. But instead, it was C15, this previously unassuming 15 carbon saturated fatty acid that was the top predictor of healthy aging dolphins.
Okay. So let's take a step back. You looked at basically all these dolphins, the ones were getting older, and you compared them, the ones that had the highest metabolic disease, and then you looked at which factor was causing that. How much of that was attributable to C15, and how much of a difference is this from other factors? If you could name some other factors you could compare it to, is there any other?
Yeah, well, absolutely. So we found, right, so these metabolomics, what it does is it ranks molecules. So it says, okay, we're going to take your top, first of all, is there a difference, right, between these two populations, healthy versus faster aging dolphins, right? And then what they showed was yes, there is a difference. And again, it had, this particular panel was unbiased. So it included lipids, carbohydrates, proteins, amino acids. It wasn't just fatty acids. And so it was this untargeted metabolomics, which is really the gift of this technology. And it showed the top 30 predictors of healthier aging dolphins. And at the top of that, so there were other molecules, to your point, it wasn't just it was only C15, but C15 was the highest ranking that was a nutrient that comes from food. So we clinged onto that because it's this is something we can actually have an intervention with. And so we gave versus a metabolite that might be more indicative of a genetic factor that's driving a healthier molecule that helps with healthier aging. We then did a feeding study where we gave dolphins fish with higher C15. So we learned that some fish had no C15, while other fish that they were eating had higher amounts of C15. So we were able to, they're given 5 different types of fish that they can eat. Some had C15, some didn't.
On purpose? On purpose you did that? That just happened?
It just happened. So we had done this experiment over, a 15, 20-year period where some dolphins were getting a low C15 diet, others were getting high. So we put a group of dolphins on a high C15 via high C15 fish diet. And that's where we saw complete resolution of things like anemia and improvement and normalization of triglycerides, cholesterol, markers of iron overload. And then and C15 was the top independent predictor specifically of resolved anemia. So once we had that, we then moved pure C15 into the lab to then say, okay, can C15 stand on its own and not just be association or, again, like you're mentioning, being mixed with other potential confounding factors.
Okay, a couple of questions. How did you even come across the idea that it could be C15? I mean, someone had to look for this. Like, why were you looking at all the fatty acids, or like, why'd you even think about that? Or just
it was luck. It was luck. I mean, so we started with a fatty acid panel because we went with a biased hypothesis, which you're not supposed to do, which is it was going to be omega-3s. And so, but we said, listen, let's just do a whole fatty acid panel and not just the different types of omega-3, omega-6s. We'll do a whole fatty acid panel. The panel that we used, if we had done the study 3 years earlier, Dr. Sanjeev, C15 wouldn't have been on the panel.
Wouldn't even on the
and
panel.
so if we had done the study 3 years earlier, this discovery would not have been made. So we're lucky that, and it's also a big reason of why C15 is like, why wasn't this discovery made before? That for a long time, C15 has been used as the reference fatty acid. So almost like the dime that you put next to other things to understand the size of everything else. It was the reference fatty acid to measure all the other fatty acids. So by default, it was excluded as being evaluated as, does it itself have benefits? So once fatty acid panels started including C15, which was again, 3 years, before we started the study, that's when it started showing up in human studies too. So there are now multiple meta-analyses. So studies of large-scale studies, prospective cohort studies, following tens of thousands of people over a decade or more, showing just like we found in the dolphins, people with higher C15 have a lower risk of developing type 2 diabetes, coronary heart disease, heart failure, and specific types of cancer. So once we saw those parallels, then we said, gosh, let's go and move this molecule into the lab and see what it can do.
Oh, so these human studies, observational studies, have existed for some time, is what you're saying. People had already been looking at this. Is that what you're saying?
Exactly. So it was in the literature and it was emerging at the same time that our studies were coming out. Again, because now fatty acid panels were just starting to include C15. So, but interestingly, those studies, they focused on the fact that even chain saturated fats like C16 and C18, so have an even number of carbons, these same studies showed that higher levels of C16 or associated with a higher risk of type 2 diabetes, fatty liver disease, heart disease, which fit our
thinking
mantra, right? Yeah. So it's like, so even though, and then what they have is at the bottom, they said, well, so exactly what we thought, higher saturated fats equals bad health. Then it was almost like a postscript. Oh, and by the way, we found that these trace odd chain fatty acids, that they were higher, interestingly associated with the opposite of better, but really let's just focus on the fact that we proved our hypothesis.
Okay. Interesting. So let's talk about naturally in humans, in our current diet, or maybe let's even talk about around the world, what type of range is there for C15? Do we know?
We do. So
concentration, percentage?
Yeah, we do. So in general, a lot of fatty acid panels use percentage. So they'll say, what percent of all the fatty acids we have in plasma or red blood cell membrane is this fatty acid. So in general, the global population is sitting around an average of about 0.2% of our fatty acids are C15.
So very relatively low. If you look at omega-3, that's like a 5% or something. Is that correct?
Exactly.
Yeah.
Great reference. Yeah. So very big difference between, which is again why people, in the fatty acid world, were like, well, it can't be important. It's present at such low levels. And, what we've now understood is that we just need enough, especially of C15 to activate certain receptors, but also to stabilize our cell membrane. C15 is a stable fatty acid that's job is to help stabilize our cell membranes against lipid peroxidation. We don't need a lot, but if we don't have that minimal amount, then the cells become fragile, more susceptible to lipid peroxidation. And in fact, what we've been able to show is that if cell membranes don't have enough C15, it causes ferroptosis, which is an entirely new form of cell death that was discovered by researchers at the Columbia University back in 2012, the same time that we reported this phenomenon in dolphins. We just didn't know that it had a name. So a nutritional
Ferroptosis, you
deficiency.
mean death by iron? What does that mean?
Yeah, that's exactly it. So what they found, it is a very distinct picture. And this is a big deal, right? Because, we go to medical school, or you go to cell biology class, and there are three ways that our cells die, right? It's apoptosis, necrosis, and autophagy. And this crew at Columbia University found a fourth way, an entirely new way our cells were dying. And when they looked under the microscope, they said the cell membranes were thickened, the cell was intact, and the mitochondria were all dead, and there was iron inside. So it was a very distinct picture.
Right.
And then the way they described it was literally the fatty acids in the cell membrane were fragile. It led to lipid peroxidation. That lipid peroxidation then combined with this free iron that was sitting in the cell results in massive reactive oxygen species, took out the mitochondria, kills the cell. So since that paper, there have been over 10,000 papers on ferroptosis, which I had never even heard of. And we now know that ferroptosis accelerates aging. It accelerates the onset and progression of type 2 diabetes, heart disease, fatty liver disease. What nobody could answer was, where did this come where did this come from? And that's where the dolphins gave us, this intel, which it popped up in dolphins because of C15 deficiencies. And we were able to fix it by putting C15 back into their diets.
So do we think that iron itself, like these people are susceptible with low C15s, giving iron supplementation may not be helpful? Is that?
Yeah, it's this huge question right now, right? So because, and you're spot on, because what happens is, we've now been able to describe the whole pathophysiology of this, what we're calling cellular fragility syndrome. We published in Metabolites last year. And what we now know is that our cells become more fragile. This includes our red blood cells. So as our red blood cells become more fragile, they get engulfed by cells within our liver called Kupffer cells. So they're these macrophages that then engulf weakened red blood cells. The problem is we have lots of weak red blood cells, right? That results in basically the corpses of these red blood cells being iron. And then you get iron overload in the liver. This is called dysmetabolic iron overload syndrome in people. And then that then feeds the beginning of ferroptosis. That then spills over systemically, and this takes time, seeds the heart, seeds the brain, seeds the pancreas. And that then leads to, if you look up iron overload and these conditions, last year, 2024, there were 1,024 papers written on iron overload. This is just starting to hit the medical and science community, and understanding the way that you're seeing it in patients would be hyperferritinemia.
Yeah, that's what I was about to just ask about. Ferritin might be a very good marker. So potentially sometimes we see people just high ferritins, but we don't really know what to really make of that. But you're saying potentially C15 should lower that ferritin then.
That's right. And that's what we saw in the dolphins, that we saw basic increases in liver enzymes. And we saw hyperferritinemia. And so when we then went back and looked at dolphin liver tissues, that's when we saw, and that's what we published in 2012, iron deposition in the liver, specifically in the Kupffer cells. So it was like we knew exactly what was happening. So what C15 is able to fix is stabilizing those red blood cells, right? Stabilizing all of our cells, but especially our red blood cells. That stops the whole pathophysiology of the disease by stabilizing the cells. Then you don't get iron overload. We're able to move this into a model, a controlled animal model that mimics this whole pathophysiology, and showed that by giving it C15, supplementing them, that it was able to completely stabilize the red blood cells, stop the iron deposition in the liver, and the downstream complications. So, but once the iron is in the liver, though, C15 can't pull it out. So it will stop the progress. What we did with the dolphins is we used phlebotomy, right? A classic old method for getting iron out of the tissues. You draw blood and the iron comes out of the tissues. But then once they had the proper C15, then, the problem was resolved.
Wow. Okay. So you were saying that the average concentration is about 0.2% of C15 in humans.
Mhm.
In dolphins, is this any different, or is there also 0.2%?
It's the same, and which is remarkable, right? And what's even more reliable is the actual concentration. So we're pushing for fatty acid panels to include the actual concentration of C15. Really, we need what's 5 micrograms per ml of C15 in our serum, in our red blood cells. That's equal to 20 micromolars, and because the percent is relative, right? And
Yes, the percent relative depends on how much total amount of fatty acids there is. But you're saying people vary in the total amount of fatty acids they have in their body?
They can. So they can vary with regard to if they're pushing, if they're taking lots of omega-3s, for example,
Right. They
right? That
they
can
skew
throw
that.
off,
Right.
the
Okay.
balance. So knowing,
Oh.
regardless of how much other fatty acids you're having, if you have that 5 micrograms per ml amount, then that would prevent nutritional deficiency syndrome. What we're working on, which is around, but right now, because most fatty acid panels only report percentages, kind of we're kind of in the ballpark of don't have a less than 0.2%. If we look at people in Sardinia who have in the high longevity zone, they have a low, and men live the longest in the world.
What's their percentages like? Do we know?
Yeah, 0.6.
Oh, wow.
So
that's interesting. So there are certain populations where you have higher rates and it's linked to increased longevity. We have that.
And a lower risk of heart disease, which there was a study done by Harvard and a whole nice conglomerate of institutions, completely separate study of a large European populations followed over time. They showed the same thing, that as you go from 0.2 to 0.6%, there's a linear decrease in risk of developing heart disease as you got higher C15 levels. So that complements nicely what they found in Sardinia. They live longer in part because they're less likely to die of heart disease. So it means that we can get to these optimal C15 levels, without supplementation, because obviously they don't have fatty 15 there. It's their lifestyle that.
What exactly in their lifestyle makes them increase their C15 levels?
It's the perfect mix that they have. So we know that if animals are grass-fed ruminants, right, that create milk, that if they are grass-fed, they have twice as much C15 in their dairy fat than if they're fed corn. So in this high longevity zone in Sardinia, they have goats and sheep that herd on the grass. Even better, multiple studies have shown that high altitude grass so when a goat or sheep eats high altitude grass, even if they're at ocean level, if they're fed this high altitude grass, they get higher C15 levels than if you're fed normal grass. So when you look at the Sardinian population, they have for the most part replaced meat with cheese, and so they have locally made cheese from these high altitude grass eating, goats and sheep. One of their cheeses is called Pecorino, which I'm a big fan of Pecorino. And Pecorino has about 50% higher C15 than other cheeses and dairy fat. So our primary source of C15, I'm not sure if I mentioned that, is dairy fat. So so they kind of have the perfect mix. In addition, they eat less than 2,000 calories a day. And they're herders, so they walk, 3 to 5 miles a day. They have it's not just eat Pecorino cheese and live past 100, right? But
grass-fed Pecorino at
high altitude, grass-fed. So it ends up, it's really fascinating population to tell us we can get to these longevity levels of C15, but they just have that perfect mix.
What's the upper limit have we seen physiologically? What number can people get to? And do we still see linear changes, or does it flatten off the curve of benefits?
It's interesting. It's the 0.6 is the highest that we've seen. This other study showing it as far as the higher C15 people had, the lower the risk of developing heart disease. That got to about 0.58. So that suggests, again, in a kind of a pre-supplement world, right, that that's about the maximum that people are going to achieve. The question is that just because no one
might be hard to do in a lifestyle way, but with the supplements, you must have done studies, I'm sure, with your supplement to see how high could you go.
Yeah. So for us, with regard to Inhumans, just within our own, our own customers and then ourselves, I am proud to say, so at Sardinia, they have between 0.4 to 0.6. So the maximum is 0.6. And so I have a 0.46. So I'm much better than the average population. And that's, it's the supplementation paired with, I don't shy away from dairy fat. I have a flat white in the morning. I have cheese, a cheese snack every day, but it's always in moderation, because the downside of the dairy fat, that we talk about is, yes, it has about 1% C15 in that dairy fat, but it has 40%.
Yeah.
It's about 40% of C16 is C18. So I still think milk is the perfect food for our young, right? It's what every mammal gets from birth. So let's not let's pay attention.
It's that way. Exactly.
It isn't the perfect food. Like no other mammal continues to eat dairy.
That's right.
For the rest of our lives. So we do need to pay attention to that. So that's why the Navy came in and said, hey, great finding. You're finding effects. Can you optimize C15 so it can be absent, so we can get reliable amounts? And at this point, it was to the dolphins. Reliable amounts to the dolphins, and make it absent of these pro-inflammatory saturated fats. And so, we made it bioavailable, free fatty acid, vegan friendly, and then just a tiny pill. So this was all developed. Military's been great at bringing new technologies to the world. This is another example of that.
So those dolphins that are living 60 years old now, what's their levels like? The ones that are they healthy 60-year-olds?
They are. They're healthy 60-year-old, which is amazing. And we've seen that, right? We've seen that with people where we all know, a 50 or 60 something year old person who is old, right? Yeah. And then we go meet an 80-year-old who is young. And we worked with Dr. Nick Short, who's the head of NIH's Longevity Consortium. And he's been in that role for over 15 years. And Nick was like, hey, Steph, I bet these dolphins can actually answer a question we've never been able to answer in humans, which is, do individuals within a same population age at a different rate? Because they don't drink, they don't smoke, they get the same healthcare. It's a very homogenous population. And indeed, we showed that dolphins, individuals within the population, had different aging rates. Aging rates were determined by their red blood cell distribution width and their hemoglobin levels, in part, but also
RDW has been definitely increased. RDW has been shown to be a risk factor, right? So
that's right.
How does C15 obviously makes an impact on that? Is that what you're saying?
Yeah, exactly. So when you talk about,
I was wondering why was RDW a risk factor, but that makes sense now.
Yeah, and so all of a sudden we have this wonderful tie to longevity, right? Because C15, like you said, RDW, it's showing up everywhere, right? As the more and red blood cell distribution with the RDW measures the amount of variability in the size of the red blood cells.
Yeah.
Now we're understanding it's like, oh, well, if your red blood cells are more fragile, they're dying faster. So you have to then work to make more new red blood cells. And that creates all this variability. It's a biomarker of aging rate, because it's a marker of the fragility of our cells. So when we put C15 back into the system, it stabilizes the red blood cells, and you see not only hemoglobin go up, but you see that RDW go down. It's so simple, right? The evolution was so simple in figuring out how humans and dolphins and elephants can live longer than mice. It's more stable cell membranes, part of what A.J. Holbert had proposed as the cell membrane pacemaker theory of aging back in 2005. He had solved it, just didn't know that there were specific fatty acids that could kind of tap into.
Okay. I know we had connected last year also a little bit talking about dogs, and I think you told me there was some research happening in that space with C15 and any update on there?
Yeah, so there've been some just a handful of studies on dogs, really interesting there, and there's an active study happening right now looking at exactly this red blood cell distribution width and cellular stability, so like osmotic fragility testing of red blood cells. Can C15 actually make these cells physically stronger? So that study's ongoing right now. But what we've learned from dogs is specifically dogs that have higher C15 have a lower risk of gastrointestinal, so a lot of gastric upset in dogs. My Maltipoo is very sensitive to whatever we feed it, and so it's a food sensitive enteropathies that dogs get. And so the dogs that have higher C15 have a lower risk of these enteropathies, which is interesting.
That's very interesting.
Yeah, there's a whole set of studies that have come out on in mouse models showing that that multiple rodent models of inflammatory bowel disease, when treated with C15, see resolution of leaky gut syndrome. So it stops the gut from being leaky, lowers inflammation, and helps repair the tissue. So they're, again, an increasing role from a whole different group looking at the role of C15 for gut health. The dogs, because dogs are so sensitive, it's popping up in that. Another really interesting finding with dogs is that, we just published with regard to Alzheimer's, that first we found that dolphins can develop the full suite of changes consistent with Alzheimer's. Only primates and cetaceans, which include dolphins, porpoises, and whales. These are the only two groups that develop the identical pathophysiology, or I should say, the histopathology of Alzheimer's disease. So these two there's just something very special. What we found is we threw C15 against 39 different targets that are for Alzheimer's treatments, like potential ways to address Alzheimer's. For us, we're looking at cognitive health, right? Not treating Alzheimer's, but ways to protect cognitive health. And C15 hit on two of these targets, pharmacologic targets. One was called MAOB. So it inhibited MAOB, monoamine.
MAOB.
Yeah, exactly. And what's really interesting, I'm getting back to the dogs here, is that studies on inhibitors of MAOB have been shown to extend longevity, not only in rodents, but in dogs. So we'll be looking more into, is this potential mechanism, in addition to strengthening cell membranes and activating AMPK, inhibiting mTOR. I mean, C15 is this pleiotropic molecule that we really were meant to have. Is this yet another mechanism, and that may be specifically relevant to humans and dogs with regard to this MAOB inhibition? So more to come on that front.
And then the part on Alzheimer's, is it related at all to glucose metabolism? Like, why is there an impact here on Alzheimer's? Like, what's the mechanism, you think?
Yeah, it's a great question. I think so. I think, we know now that Alzheimer's is that people who have metabolic syndrome, who have fatty liver disease, that it's all connected, and insulin resistance, right, being kind of a common denominator for all these conditions. Iron overload is now absolutely coming into the picture of being a common denominator, and ferroptosis between basically a disturbed metabolic system paired with that then basically feeds into and over to the brain. So we see neuroinflammation, but then paired with all of these things that are coming with iron overload and ferroptosis in the brain. So once we looked at that, so we think that's why dolphins and humans get it. There was a paper, one of the initial papers by Gunn Moore, that came out on dolphins and Alzheimer's, because they've shown this in all kinds of wild cetaceans, that Alzheimer's is present in brains of older individuals. And what their hypothesis is that Alzheimer's is not a disease of aging. It is a disease of longevity, and that animals need to live. So dogs get some portion of amyloid beta plaques, but they don't get the whole suite of changes. Yeah. And so the hypothesis is you have to live long enough, and even the theory of post-reproductive, so that how long do you live after stopping being able to have children, right? And that dictates we live long enough to basically get this disease of Alzheimer's. So that's where the dolphins come in. Again, really as a fascinating patient population. We're helping them, but they're teaching us a lot of things too.
So it's a longevity nutrient. What do you think is, I mean, you might be looking at this for future research questions, is that what's the limit here? Like, I mean, people haven't really been taking, we should talk about supplementation, but taking the C15, Fatty 15 supplementation for very long now. I mean, it's relatively, new product in the last couple of years. So we don't have understanding what will happen when people take it for 20 years, for example.
Right.
So, because we're going to hopefully be doing super doses that didn't exist, people didn't have, were difficult to attain normally. Right. So what do you think is going to happen? Like, do you think this is actually going to extend our lifespan, or this will just make the health span healthier?
I think it's more of the latter that'll have a spin-off on the former. So, C15, as we talk about in the book, is really, it checks off, and again, we worked with Dr. Shork on this, it checks off all of the boxes, what we call our 7 must-haves of a geroprotector. And a geroprotector is a molecule that slows the rate at which we age so that it can stem the onset of the diseases that kill us, right? So heart disease, Alzheimer's, cancer, and now, unfortunately, fatty liver disease and type 2 diabetes. Right. So by the goal of it, and what makes Nick so excited is that C15 can have, and again, one of the must-haves is that not only must it target the human longevity pathway, right, activating AMPK and inhibiting mTOR, targeting key hallmarks of aging. We know it helps with mitochondria, help repair mitochondria, strengthens cell membranes, decreases inflammation, but it has clinically meaningful benefits within months, right? And if you don't do that, if a molecule does not have clinically meaningful benefits within months, then it's kind of a cross your fingers and hope and pray that you live longer. But the evidence we have paired with these large population studies showing that people with higher C15 levels are less likely to get these diseases, it helps us move from association to causation. So my hope is that let's deal with the deficiency part. let's make people not deficient. Because what we're showing is younger populations are aging faster. So their aging is accelerated. Jeff Schwimmer, a pediatrician, a leader in fatty liver disease here in San Diego, he says undoubtedly kids are aging faster than their parents, and that they're getting diseases, their grandparents before their parents. That's a problem. And that's why we're the urgency is C15 deficiencies, let's fix that so that we can get longevity back on track to what it should be. And then the next is, what you're asking, which is just as exciting, right? Then can we extend our lifespan? So first, so the belief is that yes, we can, just because again, the data are so clear before us. And Nick has said, there's no other molecule, and he has seen all the longevity enhancing candidates, rapamycin, metformin, acarbose, and he says no other molecule has as robust support for being a geroprotector and a longevity molecule than C15. I mean, that it's one thing for me to say it. It's another for somebody like Nick Shork to say it.
Yeah. Okay. Let's talk about supplementation, because I think that's where obviously it's difficult to get unless you're, again, blessed with getting pecorino cheese and grass-fed and all that. And so you've made it very simple with Fatty 15 to get this small pill. And my question is that, when people look at this one small pill per day, this is incredible that it could have all these benefits. I guess it's because the percent, we already have relatively low levels and we don't need a lot. Because I think that was one of the feedback I got on our last podcast. Somebody asked, how could it be that one small amount of the C15 could cause such a huge impact? So I wouldn't mind if you could just chat about that a little bit more. And then also, what length of time does it take to get to your levels? 3 months? What are we talking about?
Right. So first, small amounts. And this is the same for micronutrients, right? So when we talk about other essential minerals and, the small amounts are often, we don't need a lot in order to maintain our baseline physiological health. So we're very lucky, and it is well established that it's these low levels that we need, which makes it easier, right, to get. There's lots of data to support that. With regard to plasma, so if we say you take C15, and I should also mention we have 100 to 200, so 1 to 2 pills a day. We have maintained a 95% monthly retention rate on Fatty15 since we launched in 2021. That kind of answers the question, right? Because people are taking this dose, and you don't have this level of retention unless people are seeing or feeling benefits. And so it tells us that this dose is appropriate for most people.
But the blood tests are showing what? Like when you must repeat blood tests after 3 months, does it increase up to 0.2? Do we get people up at, or does it double or whatever?
Yes. So the limitation of our data is that most people don't get baseline C15 levels. So we don't know how much they increase. What we do know from clinical trials, there have been 2 clinical trials with C15 supplementation, is that the lower, that for every 100 milligrams of C15 you take, it increases by 1 microgram per mil. So, and again, we want to get to 5 micrograms.
Okay.
And most of us are nobody has 0 C15. And that's because our microbiome can make some levels of C15.
Okay,
so there's there's some product. And the more fiber we eat, it helps our microbes make C15. So it's another good reason for fiber. And so it's another way that you can increase your C15 levels. But our microbiome, it's not enough in our microbiome to be able to get up and over that deficiency level. But it gives us a good head start. So that, again, that means we only want to add as much as we need to be healthy, right? And so that's where we want to start. So in order to get there, when you take Fatty15 or this pure C15 molecule ingredient, your blood levels will go up and maximize within 1 to 4 hours, and then it'll go down. And so that'll happen on the first day that you take it. What you need is to get C15 integrated into the cell membranes for these long-term benefits and into our cells and into our tissues. So that can take about 3 months for really our bodies and our tissues. Cells, red blood cells have to recycle so that they incorporate. So, and that's what studies have reliably shown, is that the best results you go to your doctor. What's even more important than higher C15 levels is, hey, my liver enzymes went down. My HbA1c is under control. I'm seeing, improvement, lower LDL cholesterol. These are things that have been shown in clinical trials, as well as, in people who are taking Fatty15. So that's what's really important. So 3 to 6 months for,
Perfect. We're gonna have a code for people to go to fatty15.com and get their supplement. Maybe before we end, what's the future directions looking like for the rest of the year?
Gosh, really exciting. We're continuing to lean into the cognitive components and benefits of C15. We are spinning out an ingredient business. So, when we talk about the importance of credibility, right? So build the science, and the science continues. And then the second is awareness. So now, people are learning and knowing about Fatty15 and C15. So we're going to keep that going based on good science. And the last is accessibility. And so Fatty15 is just a wonderful product that we've been able to bring, again, thanks to Navy's support of developing this pure ingredient. The goal of the Navy, and for us, for our team, is how do we get C15 out for all? So we're going to work on nutritional guidelines. Should parents of children that are 2 years old be told that their kids should come off whole fat milk? We want to help guide those discussions.
Right, and I didn't realize that. You're right. We sometimes, people are being told go on to skim milk and all that. That would lose their C15s.
Yeah, exactly. And so should we be doing that to our develop? So we really need to look at the science and say, can we help our youngest just by getting whole fat milk back into theirs for those who can tolerate it? How can we change agricultural practices where grass-fed animals can help increase C15 levels? Can we have dairy products report how much C15 is in their product? Because I will buy the one that has the highest C15 in it. And then the supplementation is part of the solution. And then the final part is, can we fortify foods in addition to these other activities fortify foods with C15 so that it can be, available in infant formulas and in other foods so that everybody, and for us, the whole book around longevity is how do we democratize longevity so that it's something that understanding that everybody has a right to a long and healthy life. And let's take that next step. And so that's very excited about that next step for C15.
Amazing. Well, just quick aside here. One is buying eggs that are like grass-fed from grass-fed chickens, or eating, beef that's from cows that are grass-fed. Does that have increased levels of C15?
For the chickens, I'm not sure. I don't know the answer to that. For the beef, yes. So if, for example, the meat with the highest amount of C15 in the fat of ruminants, right? Australia, something special is happening in Australia because grass-fed lamb has the highest C15. Grass-fed beef in Australia, second highest C15. If you look at moms in Australia, they have the highest level of C15 in their milk for their babies, human moms. And so there's something nice happening in Australia. And I think it's coming from the source, which is probably a lot of grass-fed herds there for the ruminants. And it feeds all the way up, which is wonderful.
Okay, awesome. What about eggs? So you don't know about eggs?
I don't know about eggs. Yeah.
I'm just wondering that we think it's a very high source. I mean, you usually see like omega-3 enriched eggs. So you think that the fat would translate over?
Yeah, maybe. I mean, it's not a reliable marker. So for sure, dairy fat is a reliable marker of how much C15 is a reliable marker of how much dairy fat we eat. And that's how it's been used for a long time. So we know that dairy fat directly translates. Other foods, that C15 is present in other foods. It's just either not reliable enough or at levels that are too low to really come through in these nutrition studies. But we want to keep every edge we can get that fiber, to get your micro there. So there's lots of little steps to help incrementally increase C15. Again, the supplement being part of the solution, but an important part that we're excited about.
Awesome. Okay, so again, I'll put the link for people to get their C15. I think we have a code for them. So I'll give that to everyone. And thank you so much for your time today. I really appreciate it.
Great. Thank you so much. It was always wonderful talking with you, Dr. Sanjeev.
Okay. Take care.
Bye.
Hey, I hope you enjoyed today's talk with Dr. Steph from Fatty15.com. Do order your supplement of the C15 longevity nutrient at Fatty15.com/Dr. Sanjeev Goel, DrSanjevGoel. I take the supplement myself, 2 capsules a day. We talked about some pretty amazing results from this nutrient. Looks like this seems to be the factor which is helping to reduce metabolic disease, prevent diabetes, prevent fatty liver disease. So super cool. We will see you next time. If you like this video, make sure to like and subscribe and share with your friends. Okay, take care. If you enjoyed today's podcast and you felt you learned something that can help you on your journey, please hit subscribe, like, and I'd love it if you'd share this with a friend who you think would also benefit and get inspired. And leave us a review. We're always looking for feedback on how to deliver more value to you. So take care. Until next time, I'm Dr. G from Peak Human Labs Podcast.